Welcome to everybody. If you may have joined us last month for our Fireflies webinar, and we are very excited to keep the invertebrate energy going this month with a talk on bees. For those of you that I have not met yet, my name is Sarah Truby, and I am a Habitat Conservation Manager with Sky Island Alliance. Today, I'm joining you from Tucson, Arizona, where the Sky Island Alliance office sits on the lands of the Tohono-O'odham, Pasquayaqui, and other indigenous people. My colleague Rick will be pasting a link in the chat for you to learn more if you'd like about that. And I see folks are still kind of coming in, so we'll vamp a little bit, and please continue to add where you're coming from. Oh, I have someone else who says they have Longhorn bachelors sleeping on their milkweed every night. I've been sharing that with Sam and Kim here. So still have some more folks coming in. So, all right, our speaker today is Dr. Kim Franklin, who is an entomologist who studies native bees. She's also the founder of the Sonoran Desert Cooperative Weed Management Area and a frequent partner with Pima County, University of Arizona, and other regionally networked conservation organizations. Also on the call is bee enthusiast and Sky Island Alliance board member Pam Rossiter, who I've had many conversations with bees about bees with. I'm sure that she'll pop up when we get to the discussion in the Q&A at the end. So today, Dr. Franklin will discuss Sonoran bee diversity and the role of native bees in our incredible ecosystems here. As a reminder, please remain muted during the talk. Type any of those questions into the chat, and at the end, we'll go through some of those because I'm sure we're going to have some great bee-related content and questions to discuss. So without further ado, let's go ahead and have you share your screen, Kim, and we can go forward.
Okay. Share and then. How's that look?
Looks great.
Okay. Okay. Thank you so much, Sarah, for that introduction, and thank you, Pam, for making this connection so that I could come to give this talk today. I'm thrilled with how many people have already shown up. This is exciting. And I am the Associate Director for Conservation at the Arizona-Sonora Desert Museum. I am trained as an entomologist. I've focused on ants and bees in my career. And I've, in the past 10 years, really shifted and spent a lot of time in the invasive species world. So I am a co-founder of the Sonoran Desert Cooperative Weed Management Area, which is run through the Desert Museum. And invasive species and pollinators, they overlap a lot. Invasive species are a threat to pollinators. They outcompete the native plants that pollinators depend on. So the work really is very much overlapping. And I'm just going to dive in here. But first, as I start this presentation, I just wanted to give a shout out to all the photographers who have provided images for this presentation. There are so many amazing macro photographers out there right now taking photos of bees and other insects. Okay, let me see, how do I advance? Okay, so the first thing I'm going to do is just tell you about the differences between honeybees and wild bees, and then I'll give you a quick tour of some of our common native bees, and then a short overview of the Desert Museum's bee research, and then I'll end with an introduction to the Tucson Bee Collaborative. Okay, so the honeybee is the bee that we are all probably most familiar with. And if you talk to people, if there's one bee that they know, it's the honeybee. It is actually a domesticated species. It was domesticated about 10,000 years ago. So it's just like cows and pigs and chickens and all a variety of crops that we grow. It's a domesticated species that makes it very useful. To us, we've selected the traits that we desire most in the honeybee. It is native. It originated in Africa and then spread into Europe and the Middle East. And so it was not present in North America until colonists brought it over in the early 1600s. So that means that honeybees were not required for pollination of any of our native plants in North America. We think of it as a really important pollinator now, and it is a very important pollinator in an agricultural setting, but none of our native plants are dependent on honeybees for pollination. The most striking difference, perhaps, between the honeybee and all of the wild bee species that I'm going to talk about in a minute here, is that the honeybee is social. It has division of labor. So in this image here, you can see the queen in the middle, and then there are a variety of worker bees that have different jobs. And so that is strikingly different than most of our wild bees. Just to give you a sense of how different honeybee, the raising of their larvae are compared to wild bees. Honeybees have nurse bees, which tend to the larvae, and a nurse bee will visit an individual larva 1,300 times daily. So they really are taking care of their infants. Everyone wants to know about Africanized honeybees. That's probably the question that I get most about honeybees. So there are about 30 subspecies of Apis mellifera. That is the honeybee, Apis mellifera. And one of those subspecies is an African subspecies. And essentially all of our honeybees here in southern Arizona are now either hybrids or this Africanized honeybee. And so it's Apis mellifera scutellata. And doesn't mean that they're bad bees. They still do, they still pollinate plants. They create honey. They are a little more aggressive than our European honeybee. So as if you were to approach a honeybee hive and it's an Africanized, they will defend that hive more quickly than, say, the European subspecies. You can't tell them apart from the European subspecies. They look nearly identical. Okay, I have to move this toolbar here. Sorry, give me a second. And we have thousands of wild bee species. And I'm using the word wild just to emphasize that these species are not domesticated. The honeybee is a domesticated species. These species are not domesticated. Worldwide, there's been about 20,000 bee species described. There's many more waiting to be described. If you just look at the US and Canada, there's about 3,600 species. And if you look at all of North America, they say about 4,000. And based on research that we're doing at the Desert Museum and some recent publications, we think that there are more than 1,000 species in the Sonoran Desert region. So we are really a global hotspot of diversity for bees. The only other part of the world that rivals that diversity are the arid regions around the Mediterranean. And that's really striking. So if you're going to look for the greatest diversity of, pick any taxa, say birds or plants or mammals, you generally are going to go to the tropics, so that this group of animals reaches their peak diversity outside of the tropics is really a special thing. I wanted to make sure I covered longhorn bees because it's been a topic between Pam and Sarah recently, and they're just really common in Tucson. You can see them if you just look inside flowers in your yard. They're not that hard to find. And this photo was taken by one of these amazing photographers, Bruce Talbert, and this article appeared in National Geographic. It was covering a peer-reviewed publication that came out in 2021, documenting the greatest species density of bees so far documented. And so this was in the San Bernardino Valley. So some of you who are local to this region probably familiar. The San Bernardino Valley is in this valley that straddles the Arizona-Sonora border in southeastern Arizona. And in 6 square miles, over the course of 10 years, they documented 500 species of bees. And so that's the greatest density. There are other studies that have documented a greater number of bee species across a much greater area. And the lead author of this study, Bob Binkley, is actually a bee systematist and will be retiring to Tucson fairly soon. So we're pretty excited about that. So I showed you the hive earlier of a honeybee. There was a queen and there were some worker bees around her. That's really strikingly different to how all of our wild bees live. So about 70% of them are going to nest in the soil. And the nest might look something like this. So there's the female bee will dig a nest after she emerges in the spring or the summer or the fall, whenever she emerges from her overwintering diapause. She will build a tunnel. It could just be a few centimeters deep. It can actually go to over a meter deep. And the architecture of these nests varies by species. And then at the end of each of these tunnels, there's a little cell, we call it a brood cell, like a little chamber. And they line those chambers with various materials that they either collect or that they produce themselves. And then they fill those chambers with pollen and nectar. So that's what this orange ball is here. It's enlarged. And people call that bee bread. So pollen is really high in protein. It is higher in protein than beef is, for instance, and it's great food for developing larvae. And she'll lay her egg on top of that ball of food, and within a week, that egg hatches, and the larva is left to develop on its own. So as in contrast to the honeybee, which has those nurse bees tending to the larvae and feeding them, this larva will develop on its own underground. So that's about 70% of our native bees. The remaining 30% nest above ground in a variety of cavities. A lot of them nest in hollow or pithy stems. So it's great to leave, dead vegetation for these bees. I think you probably have heard that, a messier yard tends to be a better habitat for insects generally, and that's true for bees. And so they nest above ground, and they create those brood cells with a variety of materials. And so this is a carpenter bee nest. And here on the far right, that's the deepest cell. It's a tunnel. So here's the deepest cell in the nest. That's a pupa. You can kind of see it looks like the bee, but it's white. Here's a ball of bee bread, but the egg didn't hatch, the larva didn't eat it, so it's still sitting there. And then here are some baby bees that are growing. And there's all sorts of cavities. I find them actually making nests in the holes in the stucco in my house. So they'll make homes in all sorts of cavities. So now I just wanted to go over some of the amazing diversity of bees that we have here, from this cactus bee over here. We have this image in the middle, and the top shows you the face of a carpenter bee, and on top of that face is sitting this tiny little perdita bee. It's one of the tiniest bees in the world. We wanted to talk about centris, a bee that Sarah has recently observed. There's just such an amazing diversity. And when you're looking at bees and you're starting to figure out what kind of bee you might be looking at, one of the best things to look at first is where you see the pollen on the bee. And so bees are collecting pollen to feed to their offspring, and only females are doing that. So you may encounter a male bee, and that male bee isn't going to have any pollen on it necessarily. So bees have either two places they tend to collect their pollen. There's some bees actually collect pollen stored in a crop, kind of like a bird has a crop, a bee has a crop, and it will hang on to the pollen, but that's rare. Most bees are collecting pollen on their hind legs, and something that we call a scopa, or a curbicula. It's a bee basket, and so they have hairs with specialized morphology that are adapted to collect the type of pollen that the bee eats. And so some bees are pretty picky eaters. In fact, many bees, especially in our region, are very picky eaters, and they will only go to certain genera, or maybe an entire family of plants, and sometimes even to just a single species, and they collect that pollen for their offspring. So bees are either collecting pollen on their hind legs, and then another group of bees, you'll see them collecting pollen on the underside of their abdomen. And so that's a really good way to first look at the division in some of the main groups of bees. And so here is a bee that I think all of you could probably see this month, because right now the barrel cactus are blooming, And barrel cactus are so easy to observe. You can just walk up to them, they're perfect height, and you can look down into those flowers, which are absolutely gorgeous. And I would say about 50% of the time you'll see a bee in there. And sometimes you won't see a bee initially because the bee has gone deep into the stamens here and it's down there drinking nectar and you don't see anything. And then boom, all of a sudden a bee appears in the flower. But so you'll see bees going down for the nectar at the base of the flower. And then you'll also see bees like this cactus bee, Diadasia rinconis, that is collecting pollen on the stamens of this cactus flower. So you'll see both things going on. Some of these cactus bees also visit globe mallow, and the globe mallow is such an easy plant to see in the urban environment. Just go look in the flowers of globe mallow in the evening. I was just telling Sarah, it's a great place to see sleeping bees. And one interesting thing about this group of bees is they often nest in aggregations. So they're not social, they're not cooperating, but they are all nesting in the same area. And that is probably one, because the resources they need are there, that they like the soil there, and also because it makes mating easier to find a mate if you're all nesting together. And so these are actually pretty common here in the Sonoran Desert. If you find the right type of soil, you'll find these nesting aggregations. They're really exciting because you'll see the bees just zooming all across the area. That's probably mostly the male bees waiting for female bees to come out and pounce on them. And often sandy soils is a good place to see these. And so here's one of our biggest bees in the area. It's Centris pallida. Some people call it a digger bee. Some people call these oil collecting bees because bees within this genus, they collect not only pollen and nectar from plants, but they collect oils, plant oils, which they use to line their brood cells where they're going to lay their eggs to waterproof them. They're big and they're hairy, so they're real easy to see, and they have beautiful eyes. So the species that's most common shown here is Centris pallida, has green eyes, has a yellow face, and has this yellow furry look on its back. And the easiest way to see them is on the Palo Verdes. And so this one's actually visiting an ironwood flower, but Palo Verdes are a great place. And there's two other species that one has red eyes and one has brown eyes, but I see them much less frequently. And here's another big charismatic bee that you'll find in downtown Tucson even. It's a carpenter bee, Xylocopa sonorina. Here's just an interesting photo of one of these bees visiting a passion flower, and you can see how the stamen has this interesting morphology such that the bee is accidentally collecting this pollen on its back. And I should mention that because a lot of people think, oh, it's great, the bees are working for us, they're doing this pollination. The bees are just collecting pollen to feed to their offspring. And the pollination is accidental, or the flowers are taking advantage of that behavior. This is the female. She's big and black, and you've probably seen her flying around. You might not have seen the male, but there are these just big, entirely yellow, fuzzy bees with green eyes, beautiful bees. This one here is actually chewing its way into that flower because it's too big to fit into it. So the term we use for this is that it's nectar robbing, as if it's not doing the job that it's supposed to do. It's robbing that nectar. And after it makes that hole in the flower, you'll see all sorts of other insects actually taking advantage of that hole. So both those bees that I just talked about, those big bees, you'll see the pollen on their legs. They're collecting pollen and pollen brushes on their legs. This next group of bees, which is in the family Megachilidae, there's a bunch of different genera, very diverse group of bees, collect pollen on the underside of their abdomens. So this is the pollen collecting hairs of this bee. So the first group that a lot of people are familiar with are leafcutter bees because bee houses have become very popular recently. So they have sharp-edged mandibles, and they cut these almost perfectly circular pieces of leaves. And so when you see a broadleaf plant in your yard and it's got lots of circle cuts out of it, it's probably a leaf cutter bee that's done that. It's not going to hurt the plant. They don't take too much of it. They're just using those leaves in the construction of their brood cells. And so they might use leaves, sometimes they use petals, and there's a bunch of other groups of bees in this family, Megachilidae. This is the genus Ashmediella. They will use different materials to construct those brood cells. Again, on this bee, you can see that where she's collecting the pollen is going to be on the underside of her. And so Ashmediella are very diverse here in the Sonoran Desert, and they're most abundant. In many parts of the country, you won't find any species of Ashmediella except the deserts of the Southwest. Also in this family is the genus Osmia, and it's a pretty well-known bee. You may have heard of this, the mason bee, because it often uses mud to sort of, as a mason would, to glue materials together to make its brood cells. And it's a beautiful iridescent blue color. And it's actually really important as a commercial pollinator. So you'll find that most of our native bees are better pollinators than honeybees. And so while honeybees are actually being transported around the country to do the pollination work, if you support native bees in the agricultural area where you're growing your crops, they can do a lot of that pollination much more efficiently and effectively than honeybees can. So this one is actually sold commercially. Okay, now moving on to a new group of bees. There are a lot of bees that get the name sweat bees. And they look there's a variety of them though. And so this is an absolutely gorgeous bee. Bees in this genus Agapostman are iridescent green. And the females are usually entirely iridescent green, and occasionally they have those striped abdomens, and the males have those striped abdomens. And so they're easy to find. They're very common. They're generalist pollinators, and you should keep your eyes open for this bee in town. And similarly, this bee is also common and abundant in the Sonoran Desert and even in the urban environment. It's Ogochlorella. But unlike the previous sweat bee, this bee is entirely green, both the males and the females. A bunch of our sweat bee species, they have these tendencies towards sociality, and so different degrees of sociality. And this one actually can shift its behavior depending on the context in which it's living. And so they find in cooler areas it tends to be more solitary, and in warmer areas it tends to be more social. And this is probably the most abundant bee out there across North America, Lasioglossum. There are a lot of different species, nearly 300 species, and the genus is a mess, and they're trying to sort out the taxonomy of this group. 3 new species were just described in January from California, but they're really, people aren't so excited about them because they're so abundant, and you find them everywhere in disturbed habitats, but also undisturbed habitats. But given their abundance, they must be important pollinators. And they're really interesting in that they exhibit the full range of social behaviors, from having that division of labor to being entirely solitary. And so I'm going to tell you a little bit about the Desert Museum's bee research. And this bee here probably makes up about 50% of the bees that we've collected in that research. And I wanted to tell you about this bee because it's also very diverse, but especially diverse here. And it has kind of specific, I think, nesting requirements, because across the sites that we've been sampling in our research, we mostly find this bee in an area, a site called Las Milpitas, which I'll tell you about. It's a urban farm that's run by the Community Food Bank. So there's 650 named species. These bees are really tiny, and they come in all sorts of colors. And I'm working with a graduate student who is trying to describe the diversity of this group, but also looking at the diversity of host plants that these bees use. And so they tend to go to very tiny little flowers. These are flowers like in the Euphorbiaceae. But that data has largely been collected from looking at observing the bees. And now the student is using, hopefully going to use eDNA. So it's just environmental DNA, looking at the different pollen that they find on these bees to figure out if they're as specific, if they're as host plant specific as they are believed to be. And then this last group of bees is a very diverse group. These bees are in all sorts of families. They're called cuckoo bees. They're kleptoparasitic bees. So these bees kind of look like wasps. They don't have a lot of hair because they are no longer collecting pollen. Instead, they sneak into the nests of other bees and they lay their egg on the pollen ball that the mother bee has provided for her own offspring. And then their larvae have a variety of adaptations. They come out first, they hatch first, and they kill the egg or the larva of the host bee, and then they consume that ball of pollen. And there's a wide diversity of these bees. A lot of them are pretty specific. They only attack a host bee in a particular genus. And so that's just a whirlwind tour of some of the bee diversity we have in our region. I'm happy to share other bees at the end of this if you have questions about particular bees. Let's see. But I wanted to save some time to talk about the bee research and that we're doing. This is a photo that I just found recently. Everyone is concerned about bee declines, and basically the data for that started with honeybees because we have lots of great data on honeybees. We can collect that data from beekeepers, and so honeybees are declining. But looking into it, we also have seen pretty significant declines in native bees, wild bees, and many other insect groups. And if we want to get into the reasons of those declines, we can at some point. Really, I think a lot of it can be summed up as the way that we grow our food, the intensification of agriculture since World War II. But in order to really look at bee declines, why there has been some, especially wild bee declines, not honeybee declines, you need long-term data. And so this, being in this hotspot of bee diversity and seeing all this news come out about honeybee declines sort of drove me in the direction of bees. I was working on ants at the time and then moved into bees. They're both in the same order, Hymenoptera. I thought, it might be a simple shift. It's much harder to identify bees than ants, it turns out. And there is a lack of long-term data. There's a lack of long-term data for probably all taxa, right? But particularly in the world of insects. And so that's why there's been some questioning of some of the studies that have come out as to whether they're really detecting declines or not. I think it's pretty well established now that they are seeing declines in many insect groups. But this was 2016, and I had gotten a grant to do some education outreach type work with the community food bank in their farm called Las Milpitas. It's a beautiful farm. Anyone can have a garden plot there, and you can also do small scale farming. It's just south of downtown. It's on the banks of the Santa Cruz, and it's just a wonderful place. And so in 2016, I started setting pan traps, which are these little cups here. They're just little cups. They're painted blue, white, and yellow, a fluorescent yellow. These are colors that have been determined to be the best colors to attract bees. And these are the same protocols that have been established as to be used nationally, so that data that different researchers studying different bees across the country will be able to better compare their data to each other. And so we set out a transect of traps in Las Milpitas. Las Milpitas here, say in downtown Tucson. And then the project grew very quickly, and we added 3 additional sites here. So one is in on the edge of Saguaro National Park here. We're also sampling at the Desert Museum, and then one way over here, which is a really interesting site near Agua Caliente Park that Pam Rossiter has been sampling for us. And since 2016, since we started, we have collected, I just checked, 26,689 bees, and they represent 70 genera. And so let me see if I tell you a little bit more about the sampling. Yeah, so we sent out these transects in the same location, and we started by sending them out every other week, and we did that for 5 years. And then we were getting, there was enough backlog that we decided to reduce the sampling frequency to just once every 4 weeks. So we cut it in half. And the next step was pinning and labeling all of these bees. So this tends to be the bottleneck when you launch a large-scale insect diversity study. Those insects tend to get stuck sitting in alcohol for years or forever. But not in this case. We have an amazing team of volunteers that I'm going to tell you about in a second, who do all this pinning and labeling and pinning and labeling and more pinning and labeling. And so they put on a label here. This is the collection label. It tells you all about where it was collected. That is identified to genus. This is the genus Lasioglossum. And then I assign it a unique identification number, and eventually it ends up in a museum collection where it will spend the rest of its life. And this is that unique number assigned from the University of Arizona Insect Collection. And so we do a lot of identification work. Well, I should say the volunteers that work with me do a lot of identification work, and identifying bees through morphology is a really challenging task. People devote their careers to learning one group of bees. You're looking at very, fine details of their morphology to try and tell these, and this is just different genera apart, not even species. And then databasing. Once a bee receives that unique identification number, it goes into a database, and all that data is digitized. And none of this work would be possible without an amazing team of volunteers through the Desert Museum, including Pam Rossiter, who's on the board here at Skyland Alliance, who made this connection so that I'm here giving you this talk today. She sampled for many years, labeled, pinned, ID'd bees, as well as all of these other people. A lot of times they find their little niche. So for instance, Ray the woman in red down there is a retired accountant, and she has kept everything so organized. We wouldn't have a program without Ray keeping us all organized. And Buzz has identified, Buzz Hoffman, that's his actual name, Buzz, has identified more bees than anybody else. And I could talk about all of these people for quite a long time, but I'm going to move on to show you a little bit of the data and save some time for questions here at the end. So this is the type of data that we can get from this study. Since we've been sampling using the same methodology for years, we can look at, this is 4 different types of sweat bees that I showed you. I showed you a bunch of different types of sweat bees, different genera, and this is their abundance in these different sites, Las Milpitas, Desert Museum, and those 2 other sites across 1 year, 2019. And you can see that their patterns in abundance change dramatically from site to site across space, but also through time. And that is one reason that is absolutely critical. If you want to look at the diversity of a site, of an area, you need to sample more than once. You need to sample a lot, because a lot of these bees spend most of their lives underground. And if we zoom in a little closer, so you can just take a closer look at some of the data. These are 2 different sites, site 1 and site 2 down here. And then the colors represent different years. And so you can, and it's just the abundance of this one swept bee in the genus Ogochlorella. So you can see how that abundance differs across the years. So if you just sampled in, say, 20, I can't see the years because 2020, you could, miss this peak in abundance that we saw in 2019. So what we've discovered from all this sampling is that in order to determine a baseline for what is like a healthy, normal population of bees and what trends might be happening with those populations, you need an enormous amount of data. I would say more than a decade of data to even begin establishing baselines for most bees. And at this point, we still have to identify all of these bees to species. They're all identified to genus, but we need the species names because the species name really unlocks everything that we know about the bee. So this is Centris pallida, and until you get a name on it, that's a rather easy one to identify, but until you get a name on it, you don't know that it is a bee that operates in very high temperatures, and it's a bee that goes to a certain variety of plants. And moreover, even though bees may be in the same genera, they can have very different life histories. And so getting to species is really important. And that's where the Tucson Bee Collaborative comes in. And so right around, I guess in 2018, I was approached by Wendy Moore and Jennifer Ketcher. Wendy Moore is the University of Arizona insect curator of the insect collection and also a faculty member in entomology there. And Jennifer Ketcher is a faculty member at Pima Community College. And Jennifer, in her classes at Pima, was using a technology called DNA barcoding to introducing the students to this technology, to introduce them to concepts in genetics. And the organisms that she was barcoding were beetles that Wendy was giving her from the University of Arizona Insect Collection. The University of Arizona Insect Collection has about 2 million specimens, but a large proportion of those are not identified to species. They knew that I was collecting bees, and they knew they were hard to identify, and so they approached me, and we started working with students to do this DNA barcoding in classrooms. And so as of today, we have barcoded about 400 different bee individuals through students. And the process goes like this. So this is not a bee. This is obviously, this is a pseudoscorpion. So we have these tiny little insects, arthropods. They look like scorpions, but they're not. They're very cute. They can't hurt you. And what you do is from whatever organism you're looking at, you extract a particular gene from that organism and you replicate it. And so you put it into this machine. This gene is this, we call it the CO1 gene. You go through a process of replication, so they have lots of copies of it, and then you send that off to a sequencing facility, and they send you back the sequence, the DNA sequence, which is a code, A, T, Gs, and Cs, and it's the order of the A, T, Gs, and Cs that let you determine, hopefully, which species you have. And then you publish that sequence in a library of DNA sequences, and the one that we're working with is the BOLD library. So this is the Barcode of Life Data System, or BOLD. The goal of BOLD, this project, is to barcode every living organism on the planet, aside from every living plant, fungus, and animal. I don't think they're doing microbes, which are a whole different ballpark, and which Pam can tell you a lot about because she is a microbial ecologist, scientist. And we publish our DNA sequences in this barcode library, and sometimes our barcode matches up with let me see, I don't have one matching up. It matches up with one that has already been identified, a bee that's been identified and exists in there, and then it's pretty easy, we get a species name. But more often than not, the bees that we're barcoding in the Sonoran Desert, there is no barcode for it existing, and so we're the first one to publish that barcode. And the task then becomes getting a morphological ID on that bee so that we can associate the DNA with the morphological ID. And then this library will be this incredible tool for identifying bees and other organisms around the world. And really being able to identify invertebrates is critical to their conservation because so much of invertebrate life has yet to be described. So the Tucson Bee Collaborative has been just this wonderful partnership. The Pima Community College students get to actually participate in authentic research. We're also involving high school students at this point and University of Arizona students. And what we've found, there's been a team that's been measuring the educational outcomes for students and found that they have an increased sense of science identity. They feel like scientists because they're contributing to something real. This will lead to persistence in STEM fields is what we hope. Also, they get introduced to this amazing diversity in this place where they live. This place-based education, I think, is so important and something that I really admire about Sky Island Alliance. I think you have all these volunteer opportunities, and through those opportunities, you do all this education, and it's very much place-based education, which I think is critical to our future. And then the U of A is getting all of these additional bees for their University of Arizona insect collection. Wendy Moores, the curator of the collection, her goal is to have a representative of every bee species in the Sonoran Desert region in the collection, represented by the bee specimen itself, by its DNA barcode, and by a high resolution image. So she's photographing these bees with an amazing camera system. And that will be a tool that will enable so much research across the globe in the future already, but in the future, especially as we fill out that barcode library for the bees of the Sonoran Desert. And me and my team of volunteers, we get bee identifications out of it. We are learning a lot about these bees. We are actually helping to build these identification tools by barcoding our bees, because the bees of the Sonoran Desert, despite being a global hotspot of diversity, are poorly studied. So we're building this tool, learning a lot about our bees, and we also get to promote our conservation mission by sharing our love for this diversity with students and with a broader audience. And so it's really been a wonderful project. I'm not going to go too much into it. I would encourage you to go to, let me see, the TucsonBeeCollaborative.org has a website, and on that website, on the main page, there is about a 10-minute piece on the Tucson Bee Collaborative that was done by Arizona Public Media. And so that's a really quick introduction. So we have students barcoding bees, and then we also have our volunteers who are having a blast here getting these bees prepared for barcoding. And I wanted to end by just, and so they are also participating in this barcoding research. They prepare the samples for the students, and then this is what it looks like when you get a barcode back. So it's just a sequence of these A, T, Gs, and Cs, the code. So this is the actual barcode of the cytochrome oxidase CO1 gene. And once you have the code, in this case, I can tell you that this bee is Lasioglossum microlipoides. We're able to put a name onto that species. So that's DNA barcoding in a nutshell. And I just wanted to end by saying that the only way that any of this is possible is that the Desert Museum has this amazing team of volunteers, and many of them have volunteered with the Bee Project and have taken on this. They're so committed to this work. They support it through their time and by learning new skills, learning how to identify these bees, and also financially by supporting us with the supplies that we need and enabling us to hire someone to help with this project. And so it's really thanks to this team of volunteers that this project exists. And I haven't been checking my time, Sarah. How did I do?
Oh, you're great. Yeah, we have a few minutes for questions, so folks can
Okay.
start typing those in the chat. Thank you so much for that. That was awesome. I loved learning where and when to see native bees. I checked before the talk and my longhorn bees were asleep, but I think they're probably gone now. I'm gonna go do a wander through my yard after this and see what else I can see. But yeah, we have some absolute great questions already. So some of them actually just relate to some of that genetic research. So I figure we'll start with those and then kind of work backwards to some of the more general ones.
Hmm.
We had a couple of different questions actually about how the genes are chosen for barcoding and how that process works. And if that's super technical, we can always send a follow-up. But yeah, if you could
Yeah.
on that.
Yeah. So they've actually chosen a single gene for all of animal life. So from whether you're barcoding a mammal or you're barcoding a bee, you're using the same CO1 gene. And that gene has been selected for a variety of reasons, but they think that it is evolving at a similar rate to speciation, at which speciation is occurring. And so there are limitations, like there may be a better gene for certain groups, but to have this consistency across all groups is really useful. And so that's the single gene that they're using.
Awesome. Fantastic. Thank you. We had a couple questions about what your favorite doc sources are for native bee identification in the region. Either a field guide, and the particular person said, I don't really feel the need to ID everything to species. I'd be tickled pink to get as
Yes.
far as family. So yeah, just something that one of us that doesn't know as much can use.
Yeah, yeah. So there are great resources, and maybe I could, if you send out a link to the recording, I could add those resources into that email.
Perfect.
There are great online resources. So I just discovered a site. It's been up for quite a while, but it's called Wild Bees of Texas. And there is some differences between the bees we have and they have, but it is such a fantastic site. I was so excited to discover this site recently, which is why it's great to always like redo these bee talks. But there are great books out there. And so there a book by Olivia Carrill. It's The Bees in Your Backyard, and I would highly recommend getting that book. It's actually, identifying bees to family isn't particularly easy. It's just more, it's easier to identify a group of bees to say a genus, and sometimes you can do that by the plants that they're visiting. But yeah, I'll send out a list of resources. The Bees in Your Backyard, I would highly recommend that. And then there's a new field guide for bees of Western North America. And then the Wild Bees of Texas is a great website.
Awesome. That's fantastic. Thank you. We had a couple of questions about diversity. Kind of what, because you talked about how it's highest in these Mediterranean regions or in our region, kind of not in the tropics. And whether that relates to floral diversity of a given region or maybe rainfall patterns, or is there what are the drivers behind that global diversity
Yeah,
pattern?
I think exactly. I do think it relates to both floral diversity, and we have an incredible floral diversity here, and rainfall patterns. The idea that has been proposed is that there is an advantage to becoming specialized here in the Sonoran Desert in areas where rainfall is very patchy in space and time. So if the bees are cued into the same cues as the plants, to when the plants are going to be blooming, plants as their resource that they need to survive and reproduce, then they have this advantage by being cued into that resource. And so when you get more specialists, and then you can less competition, and allows for more diversification. So it is that's one idea. Another idea is just that they nest in the soil, and it's a lot easier to nest in dry soils than it is to in moist soils in terms of pathogens that you might encounter. So it could be a combination of factors.
That actually relates to another question I had, which was, is there anything that I can do in my yard to protect soil nesting bees? Like if they've developed these nests, is there times of year we shouldn't be digging to plant new plants, or yeah, anything we can do to protect soil nesting bees?
Yeah, well, you can look and see if there are bees nesting before you dig, but I would say the main thing, and I think a really important thing to talk about with soil nesting bees and bee conservation generally, is that we need to provide habitat for soil nesting bees, which means not covering everything in gravel. Gravel's bad for all arthropods that are trying to nest in the soil, and most desert arthropods are trying to nest in the soil. And so making sure that, even if you live in an HOA where gravel is a requirement, if you can keep a section of your yard free of gravel, that's super helpful. Because most of our bees do nest in the soil, and bee boxes are great, but this other group of bees actually needs to be considered, their habitat requirements. And so just leaving some bare ground.
Fantastic. Love it. Okay. Awesome. And then we had a specific question once again about nesting bees, particularly where do sweat bees nest? Are they ground or cavity nesters?
They're ground nesters. For the most part, they're ground nesters. I think there's like a couple species of Ogochlorella that maybe nest above ground, but for the most part, they're ground nesters. Yeah. And you can sometimes find them nesting communally, like that aggregation I showed you.
Very cool. Also, I got a question about, do wild bees sting?
So wild bees are never going to sting you unless you like grab one. So if you like look into that barrel cactus and you grab it, and only the females can sting, males cannot sting. But they are not aggressive. There's no reason that they would sting you. Honeybees are protecting their hive. That's why they're stinging. And so they don't have that to protect. So they're very docile. And actually there's I should have put some photos of people holding these wild bees. I mean, those sleeping bees, you can edge one onto your finger and then it'll just continue sleeping there.
That sounds fun. I'm gonna go try that. We actually had a follow-up about ground nesting bees. Would mulch prevent ground nesting bees from nesting?
I think a lot of them it would, yes.
Okay. So just bare dirt. All right, I gotta go move some mulch in my yard too, so. Fantastic. Yeah. Awesome. Well, I think there's one more question that we got about bee populations, both related to the research and if there's a collection efforts are affecting bee populations at all, but then also whether there's a relationship between drought years in the desert and bee abundance.
Well, I'll start with the drought year question. So a lot of our desert bees are likely to able to spend multiple years undergrounds. A lot of them probably employ a bet hedging strategy so that some bees emerge with a rain, say they're summer bees, they're going to come out in the summer with the rainfall. So a proportion of the total pupa will actually emerge at that time when the rain comes, but some will stay underground. And so they may be underground for many years, actually. So they're sort of like desert plant seeds, they can withstand the droughts. They're adapted to it. And then I think that's a great question in terms of sampling. Is that affecting bee populations? And there is a controversy going on right now where scientists have been pushing for a national bee monitoring program that employs some standardized methods, including pan traps. And then others have been saying, we do not need to be doing that. We can sample in other ways that collect many fewer bees and will not have any impact on their populations. So I think the sampling intensity that we used here, and given the habitat that surrounded these sites where we were sampling, that we didn't have any impact on the bee populations. But even we sampled from 2016 till 2023. After that much sampling, there's no way we could tell that there's a trend upward, downward, or it's stable. And so whether or not that sampling is that useful, I think it's great to establish these patterns of phenology when these bees are active and how the diversity of bees across space and time. But now we'd like to move to more directed sampling for particular questions that we have. And one really exciting thing that's happening is that people are using eDNA, which stands for environmental DNA. And so we're building this barcode library, and we've already had some researchers come out and sample saguaro flowers. So they collected a few hundred saguaro flowers from around the Tucson Basin, washed all the DNA off of them, sequenced it all for that same CO1 gene, and then compared it to the barcode library, and they can tell us which species of bees were visiting those saguaro flowers. So I think that's a way that we'll be moving in the future, and it's very exciting. eDNA is going to have so many applications for conservation.
For sure. Yeah, awesome. We have a couple of questions about wasps versus bees, and if they're a problem for bees, or just in general, if you could speak to wasps and bees together.
Yeah, wasps, they're closely related to bees, right? But they're, so we like to say that bees are vegetarians, whereas wasps are carnivores. Wasps are great to have because they're often eating the pest insects in your gardens. And no, they're not a problem generally for bees. They're usually eating things like aphids and caterpillars and maybe some beetle larvae, or, so they're generally not attacking bees, and they're not competing with bees because wasps are feeding their offspring insect larvae and bees are feeding their offspring pollen. So they're
Gotcha. No niche competition there. So that's good.
yeah,
Okay.
yeah. And they're easy to tell apart generally because wasps are not hairy, right? Because they're not collecting pollen.
For sure. Yep. I think we'll have probably just two more questions. So one relates to honeybees impacting wild bee populations and kind of the competition there, whether there's any invasive species activity kind of there, or yeah, if there's any kind of impact between honeybee populations and wild bee populations.
Yes, I would say for sure we can say that honeybees are competitors with our native bees. They're collecting the same floral resources, nectar and pollen, and they're have been shown in some studies to be a little more aggressive. They dominate that resource. And they can spread, honeybees have a lot of parasites and pathogens affected by disease. Those diseases can be transferred to native bees. And so honeybees are definitely an issue for native bees, which is why there has been actually some more recent news coverage of that the honeybees are okay. Actually, we don't need to worry about honeybees. It would be, I think Pam sent me this cartoon where someone was comparing, birds are declining, so let's all start keeping chickens in the backyard. We want to save the birds. Let's keep chickens. It's similar with bees. You don't want to keep honeybees in your backyard if you want to save the bees.
Yeah, for sure. Awesome. And then there's the very last thing I want to ask is, are you willing to be contacted by folks directly for questions? We can include your contact information, if so, in the follow-up email.
Yeah, that is absolutely fine. And sometimes I direct those questions to different members of the bee team. The volunteers have really become experts in their own right, and so don't be offended if I direct your question to, say, Buzz Hoffman.
For sure. Oh, and then that reminds me also, someone did ask a little bit back about how do we donate? How do we support you? How do we get involved?
Yeah, well, one way would be just to contact me, especially if you're interested in donating your time or efforts to this work. And then you can make a donation to the Desert Museum through the Desert Museum's website. Yeah, those are the two primary ways. And another way to get involved is that you probably have an audience that's fairly familiar with iNaturalist. And so on iNaturalist, we have a project called the Tucson Bee Collaborative Project. And so if you're taking observations of bees, photographing bees and barrel cactus flowers this summer, now that you know that you should be looking in barrel cactus flowers, and share those on iNaturalist, and you'll be contributing to our understanding of bees.
Fantastic. Awesome. Kim, thank you so much. This was amazing.
Thank you for having me.
Yeah, thank you. For everyone else on the call, we will be sending around a recording, so you're welcome to rewatch and look at all those amazing photos of bees. You'll get an email about that shortly after the talk. And I just want to remind you to check out our website. We have a number of events coming up where you can come out and volunteer with Scott Island Alliance. And we promise to set aside time so we can look for bees at all of those events. So you will have a chance to do it right away. Thank you all so much for joining us this morning. Thank you, Kim, so much. That was excellent.
Thanks for having me.
I hope that everyone has a wonderful day. Thank you all.
Bye.